Welcome everyone. Myself Shilpi Banerjee is here to discuss about different types of libraries. Well, in the context of molecular biology and genetics, a library refers to a collection of cloned DNA or RNA fragments that represent the genetic information of a specific organism or sample. Libraries are essential resources for researchers to study, analyze and manipulate the genetic material of an organism. A genetic library is also known as a DNA library or gene library, which is nothing but a collection of cloned DNA fragments from a specific organism or sample. It represents a comprehensive or partial set of the genetic material, which is obviously DNA of a source organism. Libraries can be of different types based on the type of genetic material they contain and the purpose they serve. Number 1, genomic DNA library. A genomic DNA library contains fragments of an organism's complete set of genome. It represents the entire genetic content of an organism and is useful for studying gene organization, identifying genes and conducting genomic analysis. Moving on to the next type of library, that is cDNA library. cDNA library is nothing but complementary DNA library. It contains DNA copies of the mRNA molecules. I believe you understand mRNA. mRNA is nothing but messenger RNA molecules which is present in a specific tissue or cell type. It is generated using the reverse transcription of mRNA into complementary DNA. cDNA libraries are essential for studying gene expression and identifying genes that are active in particular tissues or cells under specific conditions. Moving on to the next type of library, that is bacterial artificial chromosome library, or in short we know it as BAC library. BAC libraries are constructed by cloning large DNA fragments whose range is typically around 100 to 300 kilobases. BAC libraries are used for genome mapping, physical mapping and large scale genome sequencing projects. Next, yeast artificial chromosome library or YAC library. YAC libraries involve cloning large DNA fragments, usually several hundred kilobases to few megabases in size, into yeast artificial chromosome. So, I believe you can clearly understand the difference between BAC library and YAC library. As mentioned earlier, BAC library size is hardly 100 to 300 kilobase pair, but yeast library, yeast artificial chromosome library has DNA fragments which is several hundred kilobases to few megabases. YAC libraries have been used for studying large genomic regions due to their extensive size and for construction of complex genomic libraries. Moving on to the next type of library, that is expression library. Expression library contains DNA fragments that are cloned into expression vectors, allowing the production of the encoded proteins. It is useful for identifying genes that produce specific proteins of interest and for functional studies of gene products. Moving on to the next type of library, that is metagenomic library. Metagenomic libraries are basically created from environmental samples. Environmental samples in what form? Such as soil, water, or even the human gut microbiome. These samples, when are collected and are constructed into library, they allow the study of collective genetic content of a microbial community. Next, epigenomic library. This is basically a specialized library which focuses on studying epigenetic modifications such as DNA methylation and histone modification. This is used to analyze epigenetic patterns and regulatory mechanisms. Next type of library in genetic engineering is genomic DNA shotgun library. Now, this type of library is created by randomly shearing genomic DNA into very small fragments and cloning them into vectors. Shotgun libraries are usually used for large scale genome sequencing projects. Next, mutant library. As the name suggests, it is very well understood that mutant library must be related with something studying of mutations, right. So, a mutant library is a collection of those DNA sequences that carry mutations in specific genes. So, it becomes pretty easy to understand and study or analyze such mutated genes. Mutant library is used to study the function of genes by analyzing the phenotypic effects of the mutations. Next, combinatorial library. Now, these are something very artificial. Artificial in what sense? They are artificially created that contain diverse set of DNA sequences, which is often used for generating libraries of peptides, antibodies, or small molecules, which is generally included in drug discovery and development. As we have now seen around 9 to 10 different type of libraries, I would like to mention here that each type of DNA library has its specific application in research, ranging from studying gene structure and function to exploring the diversity of microbial communities or discovering potential therapeutic agents. The choice of which library we have to use entirely depends on the research objectives and the specific questions being addressed to the community. DNA libraries have a wide range of applications in molecular biology, genetics, biotechnology, and other related scientific fields. Let us now see some of the key applications of DNA libraries. Number 1, gene discovery. DNA libraries, especially complementary DNA libraries, are invaluable tool for identifying and discovering new genes. They allow researchers to study the genetic content of specific tissues, developmental stages, or under certain conditions, leading to the identification of genes involved in various biological processes. Second application, gene cloning and expression. DNA libraries facilitate the cloning and expression of a specific genes of interest. By isolating DNA fragments containing the desired gene, researchers can clone and express the gene in host organism, invariably bacteria and yeast, for functional studies or protein production. Next, which is absolutely in the field of genetic engineering. DNA libraries are crucial for genetic engineering projects. Researchers and scientists can use them to find specific DNA sequences, regulatory elements or gene variants which can be manipulated or inserted into other organisms to confer desired traits or functions. Next, genome mapping. Genomic DNA libraries, specifically bacterial artificial chromosome libraries, are very much essential for genome mapping and sequencing projects. They basically help the ordered assembly of large genomic regions and in identification of genes and related regulatory elements. Next application is for genetic screening. DNA libraries are used in genetic screening to identify individuals carrying specific genetic variation which are associated with disease or traits. Screening can be done by using probe hybridization or polymerase chain reaction PCR based methods. Next application is drug discovery. Combinatorial DNA libraries are used in large scale for drug discovery to identify potential drug candidates. These libraries contain a diverse set of DNA sequences that can be translated into peptides or proteins, which are then screened for their potential therapeutic effects. Next application includes in functional genomics. DNA libraries are very much used in functional genomics for studying systematically gene functions by using techniques like RNA interference, which use DNA libraries to knock down specific gene and in turn study their effects on different cellular processes going on. Next application includes metagenomics. Metagenomic libraries, which are constructed from environmental samples, allow researchers to study the genetic diversity and functional potential of microbial communities present in various ecosystems. Genetic libraries can also be used for disease diagnosis, such as for during cancer mutations or many genetic disorders, some infectious agents and many other things. This is also helpful for analyzing and studying evolutionary studies. DNA libraries help in understanding evolutionary relationship between organisms by comparing conserved or variable DNA sequences across different species. Libraries are generally constructed through various molecular biology techniques, including DNA or RNA extraction, fragmentation, cloning into vectors which can be either plasmid or bacteriophages, and then transforming them into host cell, which is usually bacteria. Once a library is constructed, researchers can screen it to identify specific genetic sequences of interest for further analysis and study. Now, let us see how a genetic library, or let us say a DNA library, is constructed. The construction of DNA libraries involves several key steps to clone and preserve DNA fragments for various research purposes. A general overview of the process will be discussed now. The first step involves obviously extraction of DNA. So, we very well understand that DNA is extracted from the source material, which can be anything in terms of cells, tissues or an entire organism. Next, the extracted DNA is now fragmented, right. So, the next step is basically DNA fragmentation. The extracted DNA is fragmented into smaller pieces by using combination of methods or methods such as mechanical shearing, sonication or using various restriction enzymes. Fragmentation creates a diverse collection of DNA fragments of different sizes. Third step involves cloning of the DNA. The DNA which has been fragmented now is inserted into cloning vectors such as plasmid or bacteriophages. Cloning vectors act as couriers, allowing the DNA fragments to be replicated and preserved in host cells. After cloning, the next step is ligation. So, the DNA fragments and the cloned vectors are now mixed together with DNA ligase enzyme that joins the ends of DNA fragments to the end of cloning vectors, creating rDNA or recombinant DNA molecules. Next is transformation. That means, after all that cloning and ligation and everything, we need to transform them, right. So, the recombinant DNA molecules which has been produced by ligation process are now introduced into the host cells, which is usually bacteria, and it is done using a process known as transformation. Some DNA libraries may use other host organisms like yeast or mammalian cells, depending on what application it is being used for. Next is cloning and selection. Now, the transformed host cells are plated onto agar plates containing a specific antibiotic. So, only those cells that have successfully taken up vector will survive and form colonies, right. Rest might get decontaminated or may not grow. Each colony represents an individual clone carrying a specific DNA fragment from the original source. Screening. Depending on the library's purpose, researchers may use various methods to screen for specific DNA sequences or genes of interest. For example, probe hybridization, polymer chain reaction or DNA sequencing can be used to identify clones containing the desired DNA fragments. Next is storage and maintenance. Once everything is identified and verified, the individual clones are stored as glycerol stalks or as master plates. These preserved clones form the DNA library which can be accessed and used for various research applications when required. Different type of DNA libraries such as genomic DNA library, cDNA library, BAC library, expression library and whatever we have discussed till now follow similar principle during their construction, but the choice of the specific library type depends on what research objective is being followed and the type of genetic information required for the study. The construction of DNA library is the most fundamental technique in molecular biology, enabling researchers to study and manipulate DNA fragments, discover new genes, investigate gene functions and advance our understanding of genetic basis of life. DNA libraries have been instrumental in advancing our understanding of genetics, genomics and molecular biology, and they obviously be very helpful and continue to be essential tools in modern biological research. Now, let us see the concept of next generation sequencing. This concept of DNA library remains fundamental, but there are some specific modifications and adaptations to accommodate the high throughput sequencing technologies used in NGS platform. NGS allows for the parallel sequencing of millions of DNA fragments, making it a powerful tool for analyzing genomes, transcriptomes and epigenomes at a very large scale. Here is how once a DNA library is created and then it is utilized in NGS. Sequencing. Once the DNA library is prepared, it is loaded on the next gene sequencing instrument for sequencing. The library is clonally amplified, creating clusters of identical DNA fragments on the sequencing flow cell. This instrument then performs the sequencing reaction, reading the bases of DNA fragments in parallel. Data analysis. After sequencing is done, the raw data generated by the NGS instrument are processed through bioinformatics pipelines to convert the signals into DNA sequences. The sequence read are then mapped to a reference genome or assembled de novo to reconstruct the original genomic or transcriptomic sequences. Now, let us see the NGS DNA applications. Although it is almost similar, but still there are some borderline new applications which we will be seeing now. So, NGS DNA libraries have numerous applications including whole genome sequencing, exome sequencing, that is basically nothing but targeted sequencing of protein coding regions, RNA sequencing, that is sequencing of transcriptomes, chip sequencing, that is chromatin immunoprecipitation sequencing for studying protein DNA interactions, and many other epigenetic analysis. DNA libraries in NGS have revolutionized biological research and enabled the exploration of complex genomic landscapes with unprecedented depth and precision. The ability to create high quality libraries from various DNA sources and utilize the power of NGS platform has significantly advanced our understanding of genetics, gene regulation and basis of various genetic diseases. Well, while DNA libraries are valuable tools in molecular biology and genomics research, they also come with some disadvantages and limitations. We all very well know, right? Every coin has two sides. So, now let us see some of the main drawbacks of DNA libraries. First is representation bias. DNA libraries may not fully or completely represent the entire genome or transcriptome of the organism or sample from which they are constructed. Some regions might be overrepresented, while others may be underrepresented or completely absent. So, this can result from biases introduced during the DNA extraction, fragmentation or amplification steps. Next is variable in fragment length, right. So, the DNA fragmentation step during the construction of library can lead to variability in fragment lengths. So, this can impact the efficiency of sequencing, especially in paired end sequencing, where the accurate reconstruction of longer DNA fragments may be quite challenging sometimes. Next disadvantage is PCR amplification artifacts. The PCR amplification step during library preparation or construction can anytime introduce biases or artifacts. Unequal amplification of DNA fragment can completely skew or basically mismanage the representation of certain sequences, which can lead to inaccurate quantification and interpretation of the data. Next problem associated with DNA library is high input DNA requirement. So, during constructing DNA libraries for NGS typically requires a significant amount of input DNA. For some application such as single cell sequencing or working with limited clinical samples, obtaining enough starting material can be quite challenging. Next is, which is I believe is crucial step, that is cost and the complexity. however simple words we use, but this entire procedure of creating and constructing and extracting DNA consisting a library is quite complex, right. So, library preparation for NGS can be time consuming, labor intensive and expensive. It involves multiple steps, and any error or contamination during the process can affect the accuracy of the downstream sequencing results. Next is loss of information. So, during the DNA fragmentation and library preparation steps, some information about the original DNA such as long range chromatin interaction or spatial organization can be lost. So, this may limit the ability to study certain genomic features and interactions accurately. Next is limited dynamic range. DNA libraries have a limited dynamic range for quantification, which means they may not accurately represent sequences that vary greatly in abundance. High abundance sequences can saturate the sequencing, while low abundance sequences may be obscured by the background noise. Next disadvantage is bias in epigenetic studies. So, DNA library preparation for epigenetic studies, as mentioned previously, such as DNA methylation analysis, can introduce biases in the detection of modified bases, which may lead to potential misinterpretation of epigenetic patterns. Next disadvantage associated is reproducibility issues. The construction of DNA libraries involves multiple experimental steps, and variations in any of the steps involved during different experiments or laboratories can lead to issues with DNA reproducibility. Despite these disadvantages, DNA libraries still remain essential and powerful tools for various genomic and transcriptomic analysis. Researchers are continually working on improving library preparation protocols and addressing these challenges to ensure accurate and reliable results from next generation sequencing experiments. To conclude, DNA libraries are essential tools for unraveling the genetic basis of life, providing insights into biological process and facilitating advancement in medicine, biotechnology and other related scientific fields. They enable researchers to access and study the vast genetic information stored within the DNA of living organisms. With this, thank you everyone. I hope I could put some insight within you about different types of genetic libraries. On a concluding note, I believe you all must have understood that genetic libraries are indispensable part of our life. See, DNA is very complex. So, to retrieve information from it, to use information from it, for creating clones, for treating genetic disorder, everything, we need something, right. So, that is something, just like during our school days or college days, when we want to refer a subject, we need books. So, that books are kept in a library. So, similar fashion, if we want to understand about gene or DNA or the concept behind them, we need DNA libraries. So, DNA libraries are very important tools for unraveling the mystery of our life, unraveling the genetic basis of life, which provides deep insight into biological process, which helps in facilitating advancement in medicine, biotechnology, bioimaging and many other related scientific fields. So, this will actually help the researchers, scientists, doctors and that person who helps to save our life, create our life, to access, to study, to investigate and everything which basically saves life, saves genetic information, use the genetic information and create something new from it, that can be done by this DNA library. Thank you students. I hope this information, this discussion will be impactful for you about DNA library.